We are specifically focusing on Chapter 46, which covers medical parasitology.
It's a heavy chapter.
It really is. We are going to demystify the complex, the slightly terrifying, but honestly the endlessly fascinating world of human parasites. We are talking about protozoa and helminths,
Which are some of the most common, ubiquitous, and successful pathogens on planet Earth.
To truly establish the gravity of what we are dealing with here, it's important to understand that parasites rival the biggest global killers we know.
Yeah, they stand right alongside tuberculosis and HIV/AIDS in terms of their global impact and devastation.
Wow.
I mean, this isn't just a niche medical topic relegated to a specialty clinic. This is a central, load-bearing pillar of global health. It affects a huge percentage of the human population.
So by the end of this deep dive, you are going to understand the absolutely brilliant, albeit sinister, survival mechanisms of these organisms.
You'll see exactly how they evade the human immune system.
How they hijack our biology. And perhaps most importantly, you'll realize why this isn't just a quote-unquote tropical problem happening far away.
Right.
These are organisms found in wealthy nations, they're in your local daycares, and yes, they've even caused massive outbreaks at fancy luxury ski resorts.
The geographic boundaries we assume exist for these pathogens are, well, they're often a complete illusion. Human travel, global supply chains, and environmental shifts mean these organisms are constantly crossing borders.
Let's look at the sheer numbers laid out in this material, because the data from the Global Burden of Disease Study 2016, which is heavily referenced in our source material, is just jaw-dropping.
The mortality rates alone are intense.
Yeah, let me read some of these. Malaria causes over 719,000 deaths annually. Visceral leishmaniasis takes about 13,700 lives. Schistosomiasis around 10,100, and Chagas disease over 7,100.
And while those death tolls are incredibly tragic, they really only tell a fraction of the story.
How so?
Well, if we want to truly understand the impact of parasitic diseases on the human race, we have to look beyond just mortality. We have to look at morbidity.
Meaning the state of living with the disease.
Exactly. The suffering, the loss of function.
The text highlights the prevalence, the sheer number of people walking around with these infections right now. Listen to this. Ascariasis infects 800 million people.
It's a massive number.
Hookworm infects 451 million people. Trichuriasis, or whipworm, is sitting at 435 million cases. When you add up just the top 5 parasitic diseases, you are looking at over 2 billion prevalent or incident cases worldwide.
Right.
How does the medical community even begin to measure the impact of 2 billion ongoing infections?
They use a very specific public health metric, which the text explicitly discusses. It's called the DAILY, or Disability Adjusted Life Year.
Okay, break that down for us. What is a daily?
One daily essentially equals one lost year of healthy life. It is a way to measure the gap between a population's current health status and an ideal situation where everyone lives to an advanced age, completely free of disease and disability.
So it captures the fact that even if a parasite doesn't kill you outright, it steals your health, your energy, your ability to go to school or go to work.
Exactly. It quantifies the slow drain on human potential.
That makes a lot of sense.
And together, these parasitic infections cause more than 60 million dailies lost every single year.
Wow.
To put that in perspective, that exceeds the disability burden of either tuberculosis or HIV.
That is unbelievable.
When you have hundreds of millions of people carrying hookworms that are slowly draining their blood drop by drop, or millions of children burdened with massive roundworm infections that are actively competing for their daily nutrients, you aren't just looking at a medical crisis.
You are looking at a massive economic anchor dragging down entire societies. Which brings us to a major theme from the reading, the intergenerational poverty trap. This is where the biology translates into incredibly harsh realities for the communities affected.
Right. The textbook notes that parasitic diseases almost universally occur among people living in poverty.
Nearly the entire population of the world's poorest billion people is affected by either malaria or helminth infections.
Because these parasites prevent people from working at their full physical capacity. They cause severe anemia, lethargy, and chronic pain. An adult farmer with a heavy hookworm burden simply cannot produce as much food.
But for children, the impact is even more insidious and long-lasting, isn't it?
It really is. Chronic parasitic infections in childhood rob the developing body of vital iron, proteins, and vitamins. This stunts physical growth and severely impairs cognitive development.
So a child infected with schistosomiasis may struggle to concentrate or learn in school.
Exactly. And that cognitive deficit limits their future earning potential, which ensures they remain in poverty, which in turn leaves their own children vulnerable to the very same parasites living in the soil or the water.
It is a vicious, self-sustaining cycle built entirely on microscopic biology.
It really is.
Now, a crucial point for you listening right now to remember is that we have to shatter the assumption that parasites only exist somewhere else in developing nations.
That is a huge misconception.
The sources explicitly point out that parasitic diseases are also widespread among poor populations living in much wealthier nations. We are talking about the United States, European countries, and Australia.
Right. The text notes there are significant health disparities right in these regions where parasites thrive in marginalized communities.
Because of lack of sanitation, inadequate access to healthcare, and environmental exposure are not exclusive to the developing world.
Wherever infrastructure fails, or wherever poverty limits access to clean water and safe housing, these organisms will find a foothold.
So we see the massive economic and human toll this takes. But to actually fight these global killers, we have to understand what we're up against. And biologically speaking, the material breaks down the parasites into two major, very distinct armies.
Right. First, we have the protozoa.
Protozoa are unicellular eukaryotes.
They are single-celled organisms, but it is vital to distinguish them from bacteria. Bacteria are prokaryotes, they're simpler. Protozoa, being eukaryotes, are much closer to our own cells.
Meaning they have a defined nucleus housing their DNA.
Exactly. And they have complex organelles to manage their energy and structure. Because they are single-celled, they can multiply incredibly quickly inside the human host, sometimes dividing every few hours.
Which means they can cause a very rapid explosive onset of symptoms.
Right.
The text divides these protozoa based on how they move and reproduce. You have flagellates, which use whip-like tails called flagella to swim.
Then you have the amoebae, which move by extending parts of their cellular body like little feet, a process using pseudopodia.
Then there are the sporozoa, which have incredibly complex life cycles involving both sexual and asexual phases. And they are typically intracellular, meaning they physically hide inside your own cells.
And finally, the ciliates, which are covered in tiny hair-like structures that beat in unison to propel them.
That is the microscopic army. The second army is the helminths, which is the broad medical term for parasitic worms.
And unlike the single-celled protozoa, helminths are multicellular animals. They have complex tissues, nervous systems, excretory systems, and reproductive organs.
That's a huge difference.
It is. And generally speaking, a major biological difference is that most helminths do not multiply asexually inside the human body.
Meaning, if you swallow one infective worm egg, you generally get one adult worm.
Exactly. The burden of disease is directly related to how many eggs or larvae you're exposed to over time.
These worms are divided into two main categories. First, the nematodes, or roundworms. These are your classic elongated cylindrical worms with tough outer cuticles.
Then you have the platyhelminths, or flatworms.
And those flatworms are further split into trematodes, which we call flukes, and cestodes, which are the tapeworms.
The textbook gives us a really fascinating look at how scientific understanding is constantly shifting, specifically regarding how we classify these organisms.
Because taxonomy, the science of naming and categorizing life, is never static.
Right. As genetic sequencing technology improves, we sometimes discover that what we thought was a protozoan for decades is actually something else entirely.
I was reading the classification section, and the text mentions two specific groups that used to be considered protozoan parasites, but have recently been unmasked.
Yeah, this is a great plot twist.
The first is Microsporidia. Historically, they were listed with the sporozoa because they possess these highly specialized polar filaments. They literally use these filaments like tiny harpoons to pierce a host cell and inject their contents inside.
But modern genetic analysis has revealed they actually belong to the fungi kingdom.
Which is wild.
And medically, they act as opportunistic pathogens. They usually don't cause severe disease in a healthy person with a robust immune system, but they cause massive systemic issues for immunocompromised patients, such as those undergoing organ transplant chemotherapy or individuals with advanced AIDS.
The second one is Neocystis jurevecii. For the longest time, medical textbooks considered it a protozoan, but DNA analysis proved it is also a fungus.
Right. It causes a very specific, devastating type of pneumonia, interstitial plasma cell pneumonitis.
Again, primarily targeting immunosuppressed individuals. It became incredibly well known during the height of the HIV/AIDS epidemic in the 1980s.
It is wild how looking at the raw DNA just upends decades of established textbook classifications.
But pathogens do not care about our human categorization systems or our phylogenetic trees.
No, they only care about survival, evading the immune system, and finding a way to replicate.
Let's look at some of these specific battles, starting with the intestinal protozoa. We have to start with a notorious flagellate Giardia lamblia.
Ah, Giardia. It is fascinating from a mechanical perspective.
When it is in its active feeding stage inside the body, what we call the trophozoate, it lives primarily in the upper parts of the small intestine, specifically the duodenum and the jejunum.
The visual description of this trophozoate in the textbook is so memorable.
It is microscopic, only about 15 micrometers long. Under a microscope, it looks almost comical, like it has a tiny heart-shaped face.
It has 2 prominent nuclei that look like eyes, 4 pairs of flagella that it uses to swim through the intestinal fluids.
And on its ventral surface, its belly, it has this large concave sucking disc.
That sucking disc is its primary weapon and its means of survival in a very turbulent environment.
Because the small intestine is constantly moving, pushing food along.
Exactly. As the Giardia parasites pass into the gut, they use that disc to physically suction onto the intestinal villi.
The villi being the tiny, finger-like projections that line your intestines, vastly increasing the surface area for absorbing nutrients from your food.
When millions of these parasites attach to the bowel wall, they cause profound irritation and low-grade inflammation.
But more importantly, the text describes how they cause villus atrophy. They literally smother and flatten the absorptive surface of the gut.
Because the host can no longer properly absorb fats, carbohydrates, and vital nutrients, the classic pathology of giardiasis develops.
The unabsorbed fats pass straight through the digestive tract. The stools become watery, semi-solid, greasy, incredibly bulky, and profoundly foul-smelling.
It causes severe abdominal cramps, serious bloating, distension, and flatulence that can linger for months if left untreated.
But the real genius of Giardia is how it gets into your gut in the first place.
Right. As the active trophozoates move further down the colon, where the environment becomes less favorable and more dehydrated, they undergo a transformation.
They insist.
Yes. They shed their flagella, round up, and secrete a thick, highly resilient wall around themselves. They form these ellipsoid cysts that contain 4 nuclei when mature.
And these cysts are the infective form. They are passed out in the stool by the millions, and they are incredibly hardy in the outside environment.
The sources explicitly note that these cysts can survive normal water chlorination levels used in standard municipal water treatment.
This is exactly why public health officials warn you to rigorously boil or heavily filter your water when you are out camping or backpacking.
Because if a beaver, a deer, or a dog has contaminated the upstream water with their feces, those cysts can sit in the cold stream for months, completely unfazed, just waiting for a host to take a drink.
The text points out that epidemic outbreaks have occurred at luxury ski resorts right in the United States.
Which really highlights that point we made earlier.
Exactly. In those cases, the overloading of local sewage facilities during peak tourist season or the accidental cross-contamination of the municipal water supply resulted in sudden, massive outbreaks of giardiasis among tourists who never even stepped foot in the wilderness.
It's a camper's curse, but it's a resort guest's curse, too.
Moving on to another intestinal amoeba, but one that operates on a much more destructive level, Entamoeba histolytica.
The name itself is a giant red flag. Histo means tissue, and lytica means to dissolve or destroy.
Entamoeba histolytica is the causative agent of amoebiasis. It causes tens of millions of infections each year and leads to up to 100,000 deaths globally.
Like Giardia, it relies on the fecal-oral route, transmitted by ingesting its hardy cysts from contaminated water or food.
The cysts are distinct under a microscope. They have up to 4 nuclei and sometimes contain dense masses of ribonucleoprotein called chromatoid bodies.
But the active trophozoate form is where the damage really occurs.
The textbook points out a key diagnostic giveaway for this specific amoeba. If a lab technician is looking at a stool sample under a microscope, they might see these amoeboid trophozoites actively crawling around.
And inside their cytoplasm, they will literally have ingested human red blood cells.
The text calls this feature pathognomonic. If you see an amoeba that has eaten human red blood cells, you instantly know it is the highly invasive Entamoeba histolytica.
The pathology it causes when it sets up shop in the colon is brutal. When the cysts hatch, the emerging trophozoites don't just sit on the surface like Giardia, they actively invade the intestinal epithelium.
They multiply, and secrete potent proteolytic enzymes that cause lytic necrosis.
Which means they are chemically dissolving the host's intestinal cells to feed on them.
The textbook has a very specific architectural term for the type of damage it does. It produces these characteristic flask-shaped ulcers in the mucosal lining of the colon.
Right. The amoeba creates a small point of entry on the surface, burrows down via a narrow neck, and then expands laterally once it hits the softer submucosa tissue underneath.
It creates a large necrotic cavern that looks exactly like a classic wide-bottomed chemistry flask.
The destruction of this tissue causes fulminating dysentery, which is severe diarrhea filled with blood and mucus, extreme abdominal tenderness, and rapid dehydration.
But the true danger of E. histolytica is its ability to become systemic. It doesn't always stay confined to the gut.
No, it doesn't. These amoebas can actually burrow deep enough to penetrate the blood vessels lining the colon.
They enter the portal circulation, the major vein that carries blood from the intestines directly to the liver. They surf that blood flow and land straight in the liver tissue.
This is extraintestinal amoebiasis. When they reach the liver, they begin their lytic necrosis all over again, destroying liver tissue and creating massive hepatic abscesses.
The textbook disturbingly describes the contents of these abscesses. It is not your typical pus.
Normal pus is made of dead white blood cells that have fought off an infection.
But these amoebas actively destroy the white blood cells. Instead, the abscess fills with a sterile, liquefied sludge of necrotic liver tissue and digested blood.
The text describes this fluid as looking exactly like anchovy paste.
Oh man, that description stays with you.
It really does. These liver abscesses can grow massive, and if they rupture into the chest cavity or abdomen, it is a catastrophic, often fatal medical emergency.
It is a stark reminder of the sheer destructive capacity of a single-celled organism equipped with the right enzymes. Let's look at two more intestinal threats, classified as Sporozoa cryptosporidium and Cyclospora.
These are major modern threats to our water and food supplies.
Cryptosporidium typically infects the brush border of the intestinal epithelial cells. For a normal, healthy person, it causes a bout of watery diarrhea that is mild and self-limiting, usually resolving in 1 to 2 weeks.
But Cryptosporidium is severely opportunistic.
Meaning if the host's immune system is compromised, the parasite replicates unchecked.
The text notes that in patients with advanced AIDS or other severe immunosuppression, it causes intractable, unrelenting diarrhea. We are talking about massive, cholera-like fluid loss that can quickly become life-threatening.
And its environmental resilience is what makes it a public health nightmare. The infective stage, called the oocyst, has a thick wall that makes it incredibly resistant to the normal chlorine treatments used in municipal water supplies.
This specific resilience leads us to a massive historical event highlighted in the sources, the 1993 Milwaukee outbreak in the United States.
During a period of heavy spring snowmelt and rain, agricultural runoff heavily contaminated the water entering the treatment plants.
The standard chlorination did absolutely nothing to the Cryptosporidium oocysts, and a failure in the fine particulate filtration allowed them to pass right into the city's drinking water.
The result was staggering. Over 400,000 people were severely sickened.
That is nearly 500,000 people in a modern American city suffering from profound diarrhea, stomach cramps, and fever because a microscopic parasite slipped right past modern infrastructure.
It fundamentally changed how cities approach water treatment, proving that you need multiple fail-safes, including UV light or absolute microfiltration, not just chemical disinfection.
Closely related to that is Cyclospora. The life cycle is very similar, but it has a crucial difference in its transmission mechanics.
Right, when Cyclospora oocysts are passed in the stool, they are not immediately infectious. They require a period of days or even weeks in the environment to mature and become infective.
Because of that required incubation period outside the body, direct person-to-person transmission, like catching it from someone who didn't wash their hands well, is unlikely.
Instead, Cyclospora infections are heavily linked to broad waterborne and foodborne outbreaks, particularly involving complex global supply chains.
The text specifically calls out outbreaks that have been repeatedly linked to imported fresh produce. We are talking about raspberries, mesclun lettuce, and fresh basil causing widespread illness since the 1990s.
The oocysts contaminate the water used for irrigation or for washing the produce before shipping.
So when public health officials tell you to wash your produce, it isn't just about rinsing off dirt or pesticides.
It is about trying to physically dislodge microscopic cysts that could cause weeks of relapsing diarrhea, profound anorexia, and significant weight loss.
We have spent a lot of time in the gut. Let's pivot to a completely different biological battlefield.
Let's examine the parasites that target the blood and the deep tissues, the blood and tissue protozoa.
Before we dive into the blood, we need to address the one major exception in this group, a protozoan that doesn't live in the blood or the gut, but rather the urogenital tract Trichomonas vaginalis.
Trichomonas vaginalis is a flagellate, but unlike Giardia or the amoebas, it has a glaring environmentally resistant cyst stage. It only exists as an active, fragile trophozoite.
Which means it cannot survive out in the open environment. It dries out and dies almost immediately.
To survive as a species, it has to be passed directly from mucosal surface to mucosal surface through sexual intercourse. It is a highly prevalent, sexually transmitted infection.
Many infections, particularly in men, can be completely asymptomatic. But when it does cause disease, it is usually localized to the vulva, vagina, and cervix in women.
It presents with a very classic clinical picture: a profuse, frothy, yellow or cream-colored discharge accompanied by intense local burning and tenderness. In men, it can cause a mild urethritis or infect the prostate.
The most important clinical takeaway the text emphasizes here is the treatment protocol. Because one partner can be asymptomatic while harboring the active trophozoites, control of T. vaginalis requires the simultaneous treatment of both sexual partners.
If a doctor only treats the partner presenting with symptoms, the asymptomatic partner will simply pass the parasite right back during their next encounter, creating an endless, frustrating loop of reinfection.
Let's move into the true blood flagellates. The biology here operates on a level that feels almost like science fiction. Let's examine African trypanosomiasis, globally known as sleeping sickness.
This disease is caused by the Trypanosoma brucei complex, specifically the subspecies T. brucei rhodesiense and T. brucei gambiense.
They are strictly transmitted by the bite of the tsetse fly, a vector that is geographically restricted to certain regions of sub-Saharan Africa.
The progression of the disease is grim. When the infected fly bites, it injects the parasites directly into the skin and blood.
They multiply extracellularly, meaning they swim freely in the blood plasma, not inside the cells.
They spread to the lymph nodes, causing massive swelling. Eventually, they breach the blood-brain barrier and enter the central nervous system.
That invasion of the brain causes a progressive, devastating meningoencephalitis. It leads to the classic sleeping sickness syndrome.
The patient experiences severe, debilitating lethargy, a complete loss of appetite leading to profound tissue wasting, and eventually progresses to a deep, unnatural state of somnolence.
They cannot be woken up to eat or drink. It leads to coma and invariably death if left untreated.
But the truly fascinating aspect of this parasite, how it manages to survive in the human bloodstream for months or even years without the host's immune system wiping it out, is a masterclass in evolutionary evasion.
It utilizes a mechanism called antigenic variation.
This concept is incredible. Let's really break down how the immune system works to understand why this is so brilliant.
Yeah, your immune system primarily fights off invaders by recognizing the specific three-dimensional shape of a pathogen's surface proteins. These are antigens.
Once the immune system identifies the shape, it manufactures antibodies that fit that shape exactly, like a key sliding perfectly into a lock.
Once the antibodies bind to the parasite, it signals the white blood cells to destroy it.
The African trypanosomes are entirely coated in a dense, thick layer of a specific protein called the variant surface glycoprotein, or VSG.
When the human immune system detects this VSG coat, it mounts a massive, aggressive antibody response.
It floods the blood with matching antibodies and starts slaughtering the parasites by the millions. The patient experiences a high fever during this massive immune battle, and the parasite numbers in the blood plummet.
It looks like the immune system has won, but the parasite has an ace up its sleeve. The text explains that the trypanosome genome contains roughly 1,000 different distinct genes coding for completely different VSG coats.
It's an example of mosaic gene expression. Just as the host's immune system is wiping out the population, a tiny fraction of the surviving parasites spontaneously switch on a different VSG gene.
They literally shed their old protein coat and put on a brand new, genetically distinct one with a completely different three-dimensional shape.
The millions of antibodies the immune system just spent days producing are suddenly completely useless because the lock has changed.
The new variant begins to multiply rapidly. The parasite numbers surge again, causing another wave of severe fever.
The immune system has to start entirely from scratch, recognizing the new shape and building new antibodies. It clears that wave, and the parasite switches its coat again.
This cycle repeats over and over, wave after wave of parasitemia.
It constantly exhausts the host's immune resources, buying the parasite the months it needs to steadily, relentlessly invade the brain.
It is a microscopic arms race, a game of whack-a-mole, where the mole has 1,000 different disguises pre-programmed into its DNA.
Across the Atlantic, there is a different trypanosome causing a very different nightmare. Trypanosoma cruzi, the causative agent of American trypanosomiasis or Chagas disease.
T. cruzi is transmitted by the triatamine bug, commonly called the kissing bug. The nickname comes from its behavioral tendency to bite humans on the face right near the lips or the eyes, usually while the person is asleep at night.
The mechanism of infection here is particularly visceral. The bug bites the skin to take a blood meal. However, the infective forms of the parasite are not located in the bug's salivary glands. They are in the bug's hindgut.
While the bug is engorging itself on your blood, it defecates right next to the fresh bite wound.
Because the bite is locally irritating, the sleeping human reflexively scratches their face. In doing so, they unwittingly rub the infected bug feces directly into the open bite wound, or they smear it into the mucous membranes of their eye.
This specific entry route leads to a classic, highly diagnostic physical sign mentioned in the textbook Romagnus sign.
This is a severe unilateral swelling of the upper and lower eyelids on the side of the face where the feces was rubbed into the conjunctiva.
While the initial infection might cause a fever or a local lesion, the true devastating danger of Chagas disease lies in its chronic stage, which can take decades to manifest.
Unlike the African trypanosomes that stay free-floating in the blood, T. cruzi actively invades the host cells.
Once inside a cell, they transform into a small, round, non-motile stage called an amastogote, and they begin to multiply intracellularly.
They have a very strong affinity for specific tissues: the liver, the brain, and most critically, the heart muscle and the autonomic nervous system.
The text features a stark microscopic image, figure 46-5, showing dense, packed colonies of these amastogotes entirely filling the inside of human heart muscle cells.
When they invade the myocardium over years and decades, they cause chronic, low-grade inflammation and severe fibrosis scarring of the heart tissue.
They systematically destroy the conduction system of the heart, the delicate electrical wiring that keeps the heartbeat regular and coordinated.
This leads to profound, life-threatening arrhythmias, heart block, and very frequently sudden cardiac death in seemingly healthy adults.
They also target the nerve plexuses embedded in the walls of the alimentary tract. By destroying the myenteric plexus, they eliminate the nerves that control the rhythmic, coordinated squeezing of the intestines and esophagus.
The organs lose their muscle tone completely. They cannot move food or waste.
This leads to a horrific complication where the organs massively irreversibly dilate, resulting in conditions medically known as megacolon and megaesophagus.
The patient cannot swallow, or they suffer from catastrophic bowel blockages.
The human cost is immense. An estimated 7 to 8 million people harbor the parasite, primarily in Central and South America.
However, the text notes a shifting reality. Autochthonous transmission, meaning local homegrown transmission happening independently, has been documented in the southern United States, including Texas and California.
The kissing bugs live there. They carry the parasite and they are biting people.
To finish our discussion on the blood flagellates, we must examine the genus Leishmania. This parasite utilizes an infiltration strategy that is incredibly sophisticated.
Leishmania is transmitted by the bite of tiny, delicate sandflies. When the female sandfly bites, it injects the infective flagellated promastogotes deep into the skin tissue.
Normally, when a foreign invader enters the skin, your immune system's first responders, large white blood cells called macrophages, rush to the site.
Their entire job is to engulf, swallow, and use powerful acids and enzymes to digest the pathogen. A macrophage literally eats the threat to neutralize it.
But that normal immune response is exactly what Leishmania is waiting for. It allows itself to be actively engulfed by the macrophage.
Once inside the highly acidic phagolysosome, the stomach of the white blood cell, it doesn't die, it thrives.
It transforms into an amastogote, neutralizes the cell's destructive enzymes, and begins to rapidly multiply inside the very immune cell that was dispatched to kill it.
It uses the immune system as an incubator and a transport vehicle. When it multiplies enough, it physically bursts the macrophage open, releasing hundreds of new amastogotes to immediately infect neighboring macrophages.
Depending on the specific species of Leishmania, this leads to a terrifying spectrum of clinical diseases.
The first manifestation is cutaneous leishmaniasis. Species like Leishmania tropica or Leishmania major tend to stay localized near the site of the bite.
They cause dense, destructive dermal lesions, large ulcers that expand outward. These ulcers can take many months or even years to heal spontaneously, often leaving a severe, depressed circular scar. Historically, these have been known as oriental sores.
Then you have mucocutaneous leishmaniasis, caused predominantly by Leishmania brasiliensis. The text notes a terrifying progression here.
After the initial skin lesion heals, the parasites can migrate, sometimes years or decades later, to the mucosal surfaces of the nose, mouth, and palate.
They trigger a massive, destructive, hyperinflammatory response. They relentlessly erode and destroy the tissue, literally eating away the facial cartilage and the nasal septum.
In severe, untreated cases, the text notes it can lead to massive facial disfigurement and even death from asphyxiation due to airway blockage or secondary bacterial infection. The clinical picture is known in the Amazon basin as espundia.
Finally, the most severe systemic form, visceral leishmaniasis, or Kala-Azar, which means black fever in Hindi, caused primarily by Leishmania donovani.
In this form, the parasites do not stay in the skin at all. The infected macrophages migrate systemically throughout the entire reticuloendothelial system, the body's internal filtration network.
The parasites pack into the macrophages of the spleen, the liver, the lymph nodes, and deep inside the bone marrow.
The organs become massively engorged. The spleen can enlarge to the point where it fills the entire left side of the abdomen.
Because the bone marrow is compromised, the patient cannot produce enough red or white blood cells, leading to severe anemia, progressive emaciation, profound weakness, and irregular spiking fevers.
The textbook is unequivocally clear on the prognosis. Untreated cases showing symptoms of Kala-Azar are almost universally fatal. It is a highly lethal manifestation of the disease.
It is a stark biological transition we are tracking here. We start with parasites that cause uncomfortable diarrhea, and we escalate to parasites that systematically dismantle the immune system and destroy major organ systems from the inside out.
Which brings us to the most lethal and globally significant protozoa we will discuss. The organisms that target the central nervous system and the absolute heavyweight champion of parasitic diseases.
We are looking at the free-living amoebae and the sporozoa.
Let's start with the free-living amoebae. The term free-living is critical here. It means these organisms do not need a human host to complete their life cycle.
They are perfectly content hunting bacteria in soil and warm water, but when they do accidentally encounter a human, they are exceptionally lethal.
The main culprit highlighted in the material is Naegleria fowleri. It thrives in warm, freshwater lakes, ponds, sluggish rivers, and even naturally heated hot springs.
If you go swimming, diving, or water skiing and water is forcefully pushed up into your nasal cavity, you are at risk.
The amoebae enter the nasal cavity and physically attach themselves to the olfactory nerve, the nerve that controls your sense of smell.
They use that nerve as a direct highway. They follow it right through the cribriform plate, which is a porous, spongy bone located at the very top of your nasal cavity.
Once they cross that bone, they are directly inside the brain cavity.
Once inside, they cause a disease called primary amoebic meningoencephalitis, or PAM.
The text states they rapidly form massive, replicating nests of amoebae. They secrete enzymes that cause extensive, uncontrolled hemorrhage and tissue destruction, localized chiefly in the basal portions of the cerebrum and the cerebellum.
They are quite literally digesting the human brain tissue for food.
The clinical timeline is explosive. The incubation period is very short, typically 1 to 14 days. It starts with a severe frontal headache, high fever, and altered sense of smell, rapidly progressing to a stiff neck, confusion, seizures, coma, and death, usually within a week of symptoms starting.
The mortality rate is well over 95%.
The text makes a very specific modern public health connection regarding Naegleria fowleri. Outbreaks in individual cases are not just linked to swimming in lakes anymore.
Individuals have become infected and died after irrigating their nasal sinuses using a neti pot filled with unsterilized warm tap water. If the municipal water source is warm and slightly underchlorinated, the amoebae can survive in the pipes.
The rule of thumb is absolute. If you are flushing your sinuses for allergies or a cold, you must use sterile, distilled, or previously boiled water, never straight tap water.
The textbook also details another free-living amoeba, acanthamoeba. While it can cause a slow, fatal granulomatous encephalitis in immunocompromised patients, its most common danger to the general, healthy public relates directly to the eyes.
It causes severe corneal ulceration, known as acanthamoeba keratitis. The primary risk group is people who wear contact lenses.
If a user cleans their lenses with homemade or contaminated non-sterile saline solutions, or if they wear their lenses while swimming in pools or hot tubs, the amoeba gets trapped in the microscopic space between the lens and the eye.
It binds to the cornea and begins to secrete enzymes, literally eating away at the clear tissue of the eye, causing excruciating pain and potentially permanent blindness.
Now, we must transition to what is undeniably the most globally significant pathogen discussed in this entire chapter. It is the global heavyweight among all parasitic diseases, shaping human evolution and history more than almost any other infectious agent.
We're talking about the genus Plasmodium, the sporozoan parasites that cause malaria. The text states the numbers plainly, and they are devastating.
Malaria is the #1 killer among all parasitic diseases.
Looking at the 2016 data alone, there were an estimated 216 million acute cases and roughly 445,000 deaths. The vast majority of this burden falls on children under 5 in sub-Saharan Africa.
To understand why malaria is so deeply entrenched and so difficult to eradicate, we have to examine its incredibly complex life cycle. It requires 2 distinct hosts to survive: the Anopheles mosquito and the human.
Let's follow this intricate biological journey step by step. It begins when an infected female Anopheles mosquito lands on a human to take a blood meal.
As she probes the skin, she injects a tiny amount of her saliva to prevent the blood from clotting. That saliva contains the highly motile infective form of the parasite called sporozoites. They are injected directly into the human bloodstream.
But the sporozoites do not stay in the blood for very long. Within about an hour, they ride the circulation to the liver and actively invade the parenchymal cells, the main functioning cells of the liver.
This initiates the exoerythrocytic stage, meaning the stage outside the red blood cells. Inside the safety of the liver cell, the single sporozoite undergoes massive asexual multiplication.
It replicates its DNA over and over, turning into a massive schizont containing tens of thousands of tiny new parasites called merozoites.
The swollen liver cell eventually ruptures, dumping this huge, newly manufactured army of merozoites back into the bloodstream.
Now begins the erythrocytic stage, the phase that causes the actual disease. The merozoites are highly specialized to recognize and rapidly invade human red blood cells.
Once inside, they consume the cell's hemoglobin, growing from a small ring shape into a larger trophozoite, and finally dividing again to form a mature schizont containing a new brood of 10 to 36 merozoites.
And here's the critical pathological mechanism: they multiply in a highly synchronized fashion. Millions of infected red blood cells across the entire body reach the bursting point at the exact same time.
They all rupture simultaneously, releasing a massive wave of toxic cellular debris, parasite waste products, and a fresh brood of merozoites directly into the blood plasma.
This synchronized explosive bursting is exactly what causes the classic ancient symptoms of malaria. The text notes that depending on the species, this rupture happens on a strict biological clock.
Every 48 hours for species like Plasmodium vivax and Plasmodium falciparum, or every 72 hours for Plasmodium malaria, the patient experiences a severe fever paroxysm.
It is a 3-stage traumatic event. It begins with an intense, bone-rattling, shaking chill that can last for an hour. The patient feels freezing cold despite the environment.
Then the temperature spikes dramatically, frequently reaching 40 degrees Celsius. That's 104 degrees Fahrenheit. The patient burns up as the immune system reacts to the massive amount of debris.
Finally, as the fever breaks, the patient experiences profuse, drenching sweats, leaving them exhausted. They recover, only for the cycle to repeat exactly 48 or 72 hours later as the next brood bursts.
The constant systemic destruction of red blood cells leads to severe normocytic anemia. The blood simply cannot carry enough oxygen to support the body's tissues.
But there is a crucial biological detail the textbook highlights regarding the different species of Plasmodium, specifically a hidden danger that can cause the disease to return long after the patient thinks they are cured.
If a patient is infected with Plasmodium vivax or Plasmodium ovale, they face the danger of the hypnozoite.
When the initial sporozoites of vivax or ovale enter the liver, a certain percentage of them do not immediately begin multiplying. Instead, they enter a state of deep metabolic hibernation.
They become hypnozoites, or sleeping forms. They can sit quietly inside a liver cell, completely undetected by the immune system, for months or even up to 5 years after that first mosquito bite.
Long after the active parasites have been cleared from the blood by medication, these hypnozoites can suddenly reactivate, multiply, and dump a fresh wave of merozoites into the blood, triggering a full-blown malarial relapse.
This biological quirk fundamentally changes how doctors must treat the infection. If you are treating a patient with Vivax or Oval, you cannot just prescribe a drug that kills the parasites in the blood, like chloroquine.
You have to administer a second, highly specific drug like primaquine, which has the ability to penetrate the liver tissue and actively seek out and destroy the dormant hypnozoites. If you skip that step, the patient is almost guaranteed to relapse.
Contrast that complex liver stage with the most dangerous species, Plasmodium falciparum. Falciparum does not form hypnozoites in the liver. It has no dormant stage. Once it leaves the liver, it is in the blood for good.
Yet it is by far the most pathogenic, aggressive, and deadly of all the malaria species.
The reason falciparum is so much deadlier comes down to a terrifying mechanical alteration it makes to the physical properties of your red blood cells.
As the parasite matures inside the red blood cell, it exports specific, highly sticky parasite proteins known as PFE-MP1 and embeds them directly into the outer surface of the red blood cell membrane.
It turns a normally smooth, slippery red blood cell into a microscopic ball of Velcro.
Red blood cells are designed to flex and slide smoothly through the tiniest capillaries in the body. But these modified, sticky falciparum cells adhere tightly to the endothelial lining of the blood vessels, and they stick to other uninfected red blood cells.
They clump together in massive numbers. This process is called sequestration. These sticky masses physically plug the deep capillary beds of the body's major organs.
This leads to profound obstruction of blood flow, microscopic thrombosis, and severe local ischemia, meaning the surrounding tissues are completely starved of oxygen because fresh blood cannot get past the blockage.
When this mechanical blockage happens in the deep capillary beds of the brain, it causes cerebral malaria. The brain tissue swells, oxygen levels plummet, and the patient rapidly progresses into seizures, coma, and very often death.
When it happens in the kidneys, the massive destruction of red blood cells and the compromised blood flow causes acute renal failure.
The urine turns incredibly dark from the sheer volume of excreted hemoglobin, leading to a historic, highly fatal complication known as blackwater fever.
Falciparum malaria is an acute medical emergency because its ability to clog the vasculature can shut down multiple organ systems within hours.
The final two blood and tissue sporozoa we need to discuss are Toxoplasma gondii and Babesia microti. Toxoplasma is an incredibly successful parasite with a fascinating, heavily studied link to domestic animals.
Toxoplasma gondii has a truly worldwide distribution, and it is capable of infecting a vast range of warm-blooded animals, including humans, birds, and livestock. However, its life cycle has a very specific bottleneck.
The definitive host, the absolute only place where the parasite can undergo its sexual reproductive stage and produce environmentally resistant oocysts, is the feline family. Cats are the biological engine for Toxoplasma.
The cycle typically involves a cat eating an infected rodent. The parasite reproduces in the cat's intestine, and the cat sheds millions of highly infectious oocysts in its feces for a few weeks.
These oocysts contaminate the soil. Humans can acquire the infection by accidentally ingesting these microscopic oocysts, for example, by cleaning a heavily contaminated litter box or by working in a garden where a neighborhood cat has defecated and then touching their mouth.
But the text emphasizes another equally important route of human infection, eating undercooked meat.
If an intermediate host, like a pig or a sheep, grazes on pasture contaminated with cat feces, they ingest the oocysts.
The parasites hatch, migrate out of the animal's gut, and form microscopic dormant tissue cysts deep inside the animal's skeletal muscle and brain.
If a human then buys that meat, say a cut of pork or lamb, and eats it without cooking it to a high enough temperature to kill the cysts, they become infected.
For the vast majority of normal, healthy adults, acquiring toxoplasma is a relatively minor event. The immune system quickly walls off the parasites into dormant cysts, and the person might experience mild, fleeting flu-like symptoms, or absolutely no symptoms at all.
They will carry those dormant cysts for the rest of their lives without issue. But there is one specific demographic where this parasite poses a grave, catastrophic danger.
Pregnant women. If a non-immune woman, meaning she has never been exposed to toxoplasma before, acquires the infection for the very first time while she is pregnant, the active dividing parasites circulate in her blood.
They have the ability to cross the placental barrier and directly infect the developing fetus.
This results in congenital toxoplasmosis, and the pathology is devastating for the developing tissue.
If the mother is infected during the first trimester, when fetal development is most critical, it usually results in spontaneous abortion, stillbirth, or massive, life-incompatible anomalies.
If infected later in the pregnancy, the parasite targets the central nervous system of the fetus. It causes severe neurologic destruction, leading to hydrocephaly fluid buildup in the brain, microcephaly, and severe psychomotor retardation.
It is also a major cause of congenital blindness, as the parasite actively destroys the tissue of the retina, a condition called chorioretinitis.
This precise biological mechanism is exactly why obstetricians strongly advise pregnant women to avoid cleaning cat litter boxes, to wear gloves while gardening, and to ensure that all meat they consume is thoroughly, rigorously cooked to a safe internal temperature. The stakes are simply too high.
Finally, for the blood sporozoa, we have Babesia microti. The text notes this is a tick-borne infection primarily found in the coastal regions of the northeastern United States and parts of Europe.
Babesiosis is fascinating from a diagnostic perspective because it heavily mimics malaria. The parasites are transmitted by the bite of the Ixodes tick, the same tick that carries Lyme disease.
Once in the blood, Babesia parasites infect the red blood cells and destroy them, causing hemolytic anemia, spiking fevers, chills, and severe jaundice as the liver tries to process the destroyed blood cells.
It is particularly severe and often fatal in the elderly, in patients who have had their spleens removed, and in people with advanced AIDS.
When a hematologist is examining a blood smear under a microscope to confirm the diagnosis, they are looking for a very distinct, highly diagnostic visual cue.
As the Babesia parasite divides inside the red blood cell, it often forms a tetrad. It splits into 4 tiny rings arranged in a perfect cross shape.
It looks exactly like a microscopic Maltese cross embedded inside the red blood cell. That specific geometric shape instantly differentiates it from malaria.
We've covered a massive amount of ground exploring the complex, single-celled protozoa. It is time to drastically scale up in size and complexity. We are moving from the microscopic hitchhikers to the giant worms.
We are entering the realm of the intestinal helminths. We are leaving organisms measured in micrometers and discussing parasites you can easily hold in your hands. Organisms that have complex nervous systems and tough muscular bodies. Let's begin with the nematodes, commonly known as the roundworms.
To give a clear picture of what we're dealing with, let's look at their basic anatomy. Nematodes have that classic archetypal worm shape. They're cylindrical, elongated, and completely unsegmented. They look like smooth tubes, tapered at both ends, almost like a piece of spaghetti or a thick needle.
They're covered in a tough, complex, protective outer cuticle that they actually have to shed or molt several times as they grow from larvae to adults.
Internally, they're quite advanced. They possess a complete straight digestive system with a mouth opening at the anterior end and an anus at the posterior end.
They maintain their body shape using a hydrostatic skeleton fluid under high pressure inside their body cavity. And unlike many other parasites, nematodes generally have highly developed, distinct, separate sexes. You have clearly identifiable male and female worms that must mate to reproduce.
The first nematode we need to examine is an absolute monster in terms of both size and prevalence. Ascaris lumbricoides, the giant human roundworm.
The physical dimensions of this parasite, considering it lives inside the human small intestine, are shocking. Adult female Ascaris worms routinely measure 20 to 50 centimeters in length.
That is nearly half a meter or over 1.5 feet long and as thick as a standard pencil. The males are slightly smaller, usually 15 to 30 centimeters, and can be identified by a characteristic sharp curl at their tail end.
The sheer size is disturbing, but the life cycle, the physical journey this worm undertakes inside the human body, is one of the most bizarre and punishing examples of parasite evolution.
It starts simply enough. A human accidentally ingests microscopic Ascaris eggs from soil that has been contaminated with human feces, perhaps on unwashed vegetables or dirty hands. The eggs travel down to the small intestine, and the tiny microscopic larvae hatch.
Logically, you would expect those larvae to just stay in the nutrient-rich environment of the small intestine, feed, and grow into those 50-centimeter adults. But that is not what happens.
The newly hatched larvae actively bore through the mucosal wall of the intestine and push their way into the tiny venules of the portal circulatory system.
They enter the bloodstream. They are carried by the blood flow directly through the liver tissue, into the inferior vena cava, into the right side of the human heart, and then they are pumped forcefully into the pulmonary artery, straight into capillary beds of the lungs.
At this stage, the larvae are growing rapidly and demand high levels of oxygen. They become too large to pass through the tiny capillaries of the lungs, so they physically rupture the blood vessels and break out into the alveoli, the delicate microscopic air sacs where oxygen exchange occurs.
Having thousands of microscopic worms tearing through the lung tissue triggers a massive, severe inflammatory response in the host. The patient develops a condition called Loeffler's syndrome.
They present with a harsh, hacking cough, a massive spike in eosinophils, which are the white blood cells specialized to fight parasites, and thick pulmonary infiltrates that look like severe pneumonia on an X-ray. The host is coughing violently because their lung tissue is literally filled with migrating worms.
The worms use that coughing reflex to their advantage. They crawl up the bronchi, up the trachea, right to the back of the throat, riding the mucociliary escalator.
When they reach the epiglottis, the host reflexively swallows them back down. They survive the stomach acid a second time, return to the small intestine, and only then do they finally settle down to mature into massive adults.
It is an incredible, highly destructive detour. Why would evolution favor such an arduous, dangerous migration just to end up exactly where they started?
Biologists theorize it ensures the larvae have access to highly oxygenated blood during a critical, fast-growing developmental phase before they are tough enough to survive the low-oxygen, highly competitive environment of the adult gut.
Once they mature, they mate, and a single female Ascaris can release an astonishing 200,000 eggs per day into the host's feces, perpetuating the cycle wherever sanitation is poor.
Because of their sheer physical size, a heavy infection of Ascaris poses severe mechanical dangers. If a child harbors dozens or even hundreds of these giant worms, they can physically tangle together into a massive knot, causing a complete, life-threatening mechanical bowel obstruction that requires emergency surgery.
The text also notes they are highly sensitive to their environment. If the host develops a high fever or is given certain anesthetics, the worms become agitated and tend to wander. They can crawl up into the biliary tree, blocking the bile ducts, or force their way into the appendix, causing acute appendicitis.
Next on the list are the human hookworms, Ancylostoma duodenale and Notator americanus. These are much smaller worms, only about 1 centimeter long, but they are incredibly damaging due to their unique method of infection and their brutal feeding mechanics.
The hookworm life cycle does not start by accidentally swallowing a dormant egg. The eggs are passed in the feces and actually hatch outside the body in warm, moist, shaded soil.
The larvae go through a couple of molts over a few days until they become highly motile, infected falciform larvae. They sit in the upper layers of the soil or on blades of damp grass, just waiting.
When an unsuspecting human walks by barefoot, or a farmer works the soil with their bare hands, the larvae detect the body heat and chemical signals. They do not wait to be eaten. They physically, actively penetrate the intact skin of the human feet or ankles.
The initial entry causes a local allergic reaction known as ground itch, characterized by intense itching, redness, and a vesicular rash exactly at the site of penetration. From there, the larvae undertake the exact same destructive blood-to-lung migration that Ascaris does.
They travel to the lungs, are cocked up, and swallowed down to the small intestine.
This is where their true damage begins. The text features a vivid, almost terrifying microscopic image, figure 4615, showing the specialized mouthparts of the hookworm.
It does not have a simple tube opening. It has a robust buccal capsule equipped with hard, sharp, chitinous cutting plates, or teeth.
It uses these plates to bite a deep chunk out of the intestinal mucosa. It clamps tightly onto the highly vascularized intestinal villi, secretes powerful anticoagulants to keep the blood flowing, and feeds directly on the host's arterial blood and tissue fluids.
A single hookworm might only ingest a tiny fraction of a milliliter of blood per day. But people living in endemic areas are routinely infected with hundreds, sometimes thousands of these worms.
This chronic, relentless daily blood loss, which often leaks into the bowel even after the worm detaches to find a new biting site, is the root cause of the profound, widespread iron deficiency anemia associated with hookworm infection.
This anemia leads to severe fatigue, pallor, heart murmurs, and in children, significant delays in physical and mental development, contributing heavily to the global poverty trap we discussed at the beginning of the dive.
It is a microscopic organism draining the energy out of 451 million people worldwide, drop by drop. Now let's examine a nematode that possesses a set of evolutionary adaptations that make it uniquely dangerous and difficult to eradicate: Strongyloides stercoralis, the human threadworm.
Strongyloides is exceptional among the helminths for two massive biological reasons. First, the adult parasitic females that live embedded in the human intestinal mucosa are parthenogenetic.
This means they do not require a male worm to fertilize them. They can produce and lay viable, fully functional eggs all on their own.
The second and far more dangerous adaptation is the phenomenon of autoinfection. For almost every other nematode, the eggs or the early-stage larva must leave the human body in the feces.
They require a period of development in the outside soil before they can infect a new host or reinfect the same host. The cycle is broken if sanitation is good.
But the larvae of strongyloids can rapidly develop into their aggressive, tissue-penetrating filariform stage right inside the human colon. They hatch from the eggs laid in the mucosa, and before they are swept out with the feces, they actively penetrate the wall of the lower intestine, or the perianal skin.
They re-enter the host's bloodstream, perform the lung migration, and return to the gut to mature into new adults. They complete their entire life cycle entirely within the same host.
This mechanism means a single exposure can lead to a lifelong infection. A person can harbor a strongyloids infection for 40 or 50 years, long after they have left an endemic area, because the worms are constantly quietly reinfecting them from the inside.
While this chronic autoinfection is usually kept in check at a low level by a healthy immune system, there is a severe, life-threatening medical warning associated with this parasite.
If a patient with an unrecognized chronic strongyloids infection is given powerful immunosuppressive drugs, specifically high doses of corticosteroids for asthma and autoimmune disease, or prior to an organ transplant, the parasite's internal autoinfection cycle loses all immune restraint. It explodes into overdrive.
It produces a catastrophic condition called hyperinfection syndrome. Tens of thousands of aggressively migrating larvae suddenly tear through the wall of the gut simultaneously.
And as they punch through the colon, they carry a massive load of deadly enteric bacteria on their surface directly into the sterile bloodstream. This leads to overwhelming, often fatal gram-negative sepsis, meningitis, and massive pulmonary hemorrhage.
It is a critical, absolute requirement for doctors to screen for strongyloids before prescribing heavy steroids to any patient who has ever lived in an endemic area.
It is a terrifying example of a parasite exploiting a modern medical intervention. Let's look at a nematode that is far less lethal but incredibly pervasive and highly irritating, Enterobius formicularis, the human pinworm.
Pinworms are perhaps the most common helminthic infection in the United States and Europe, and they primarily, famously, affect young children. They are tiny worms. The adult females are only about 10 millimeters long, about the size of a staple.
The defining behavior of the pinworm, and the reason it is so successful at spreading, is its highly specific egg-laying strategy. The adult worms live peacefully in the cecum and the colon, but they do not lay their eggs there.
At night, when the host is sleeping and the body temperature drops slightly, the gravid female worm actively migrates down the colon, crawls completely out of the anus, and deposits up to 20,000 sticky eggs directly onto the perianal folds of the skin.
These eggs are highly immunogenic, meaning they trigger a severe, localized hypersensitivity reaction in the skin. The primary overwhelming symptom of a pinworm infection is intense perianal pruritus, uncontrollable, maddening itching, specifically occurring at night.
The child naturally scratches the intensely itchy area in their sleep. The microscopic, sticky eggs become lodged under the child's fingernails. The next day, the child touches their toys, their bedding, their own mouth, or other children at daycare.
The eggs are remarkably resilient in the indoor environment, surviving in dust and on surfaces for days. They are ingested, they hatch in the stomach, and the cycle completes within a matter of hours. It spreads through households like wildfire.
The diagnostic method the text describes for identifying pinworms is wonderfully low-tech and specific. It's the classic Scotch tape test.
Because the female lays her eggs on the outside of the body, standard stool examinations that look for eggs mixed in the feces will almost always come back negative, missing the infection entirely.
Instead, pediatricians instruct parents to use the tape test. First thing in the morning, before the child bathes or has a bowel movement, you firmly press a piece of clear, transparent Scotch tape directly against the perianal skin. The sticky eggs adhere to the tape.
You stick that tape flat onto a glass microscope slide. Under the scope, the eggs are highly distinctive. They are oval but flattened on one side, looking exactly like tiny microscopic footballs that have been slightly deflated.
We have one more highly significant nematode to discuss, one that bridges the gap between the intestine and the deep tissues. Trichinella spiralis.
Trichinellosis is a zoonotic disease. Its natural life cycle is entirely maintained within animal populations, specifically carnivores and omnivores. It cycles constantly between pigs, rats, bears, wild boars, and walruses.
Humans enter the cycle strictly as accidental dead-end hosts. We acquire the infection by eating raw or improperly undercooked meat that contains the encysted larvae of the worm.
Historically, this was heavily linked to commercial pork. Today, with better agricultural regulations, outbreaks are frequently tied to hunters making homemade sausage from wild game, specifically bear or wild boar, and not cooking it thoroughly.
You eat the infected meat, The stomach acid digests the meat but leaves the tough cysts intact, and the larvae emerge in your small intestine. They rapidly mature into adults and mate within a few days.
But the female trichinella worms have a unique reproductive strategy. They do not lay eggs that pass out in the feces. Instead, they give birth to live, highly motile larvae directly into the mucosal wall of the human intestine.
These tiny larvae enter the lymphatic system and the bloodstream, circulating throughout the entire body. However, they are highly selective. They only seek out and survive in highly active, oxygen-rich, striated skeletal muscle tissue.
The preferred targets are the diaphragm, used for breathing, the masseter muscles of the jaw, used for chewing, the tongue, the extraocular muscles of the eye, and the large muscles of the arms and legs.
What they do when they penetrate those individual muscle cells is a marvel of biological hijacking. The larva enters the cell and fundamentally manipulates the host's DNA.
It forces the muscle cell to stop producing contractile proteins. It stops functioning as a muscle. Instead, the parasite forces the cell to enlarge to secrete a protective collagen capsule around the worm, and it actually upregulates factors like VEGF, vascular endothelial growth factor, to stimulate the growth of a dense capillary network completely surrounding the infected cell.
It biologically forces the human body to build a custom, highly vascularized life support system. The text calls this manipulated structure a nurse cell.
The worm sits curled up inside this nurse cell, safe from the immune system, feeding on the nutrients supplied by the new blood vessels, remaining viable for years or even decades. The mass invasion of these muscles causes severe, acute myalgia, intense muscle pain, facial swelling, fever, and profound eosinophilia.
It is a brilliant, terrifying example of a parasite rewriting host cellular machinery for its own benefit. Let's wrap up our discussion on the intestinal helminths by shifting entirely away from the cylindrical roundworms to the flatworms, specifically the cystodes or tapeworms.
Tapeworms look and operate fundamentally differently than nematodes. They are incredibly flat, ribbon-like creatures. Their anatomy is highly specialized for their environment.
At the very front, they have a tiny head structure called a scolex. The scolex is purely an anchoring device. Depending on the species, it is equipped with muscular suckers or rings of sharp, chitinous hooks that it uses to embed itself tightly into the intestinal wall so it doesn't get swept away.
Behind the scolex, the worm consists of a continuously growing chain of independent rectangular segments called proglottids.
The most profound biological difference is that tapeworms have absolutely no digestive system. They have no mouth, no stomach, no intestines.
Because they live their entire adult lives completely bathed in the rich, pre-digested food of the host's small intestine, they don't need to digest anything. They simply absorb carbohydrates and amino acids directly through their specialized skin called the tegument.
Each proglottid segment is essentially an independent hermaphroditic reproductive factory. As the segments are pushed further down the chain, they mature, mate with themselves or neighboring segments, and fill entirely with tens of thousands of eggs. The mature segments at the end of the tail break off and pass out in the feces.
The text highlights a specific massive tapeworm called the broadfish tapeworm, Diphyllobothrium latum. Its life cycle involves tiny crustaceans, freshwater fish, and humans. Humans become infected by eating raw or undercooked infected freshwater fish like salmon or pike.
The size of this worm is astounding. Inside the human gut, Diphyllobothrium latum can grow up to 10 meters long. That is over 30 feet of flatworm coiled up inside your digestive tract. And it causes a very specific, unique pathology.
Most tapeworms are relatively asymptomatic, perhaps causing slight abdominal discomfort. But the fish tapeworm has an unusually high, aggressive affinity for absorbing vitamin B12 from the human gut. It is incredibly efficient at it.
It steals so much of the vitamin that it can cause the human host to develop a severe vitamin B12 deficiency. Since vitamin B12 is critical for the proper formation of red blood cells and nerve function, this deficiency leads to a specific severe condition called megaloblastic or pernicious anemia.
The patient develops huge dysfunctional red blood cells, profound weakness, and potential neurological damage. The parasite is literally outcompeting the human host for essential micronutrients.
This concept of worms causing systemic damage leads perfectly into our next category. We are moving beyond the gut entirely to examine the worms that invade the body's internal infrastructure.
We are entering the domain of the blood and tissue helminths. These are worms that don't stay in the digestive tract. They actively invade the skin, the deep tissues, the eyes, and the lymphatic system. Let's start with a group called the filarial nematodes. These are incredibly thin, thread-like worms that rely entirely on blood-sucking insect vectors for transmission.
The most globally significant disease in this category is lymphatic filariasis, caused primarily by the worms Wuchereria bancrofti and Brugia malae.
The vector here is the mosquito. When an infected mosquito bites, it deposits highly motile microscopic larvae onto the skin, which actively crawl into the bite wound. But instead of entering the bloodstream or the gut, these larvae specifically seek out and migrate deep into the human lymphatic system.
The lymphatic system is a crucial network of delicate vessels and nodes that runs parallel to the circulatory system. Its primary job is to drain excess interstitial fluid from the tissues and return it to the blood, while also serving as a highway for immune cells.
The filarial larvae mature into thin adult worms. The females can be up to 10 centimeters long, and they set up permanent residence directly inside the afferent lymphatic vessels, typically in the lower extremities or the groin.
The pathology is caused by a combination of the physical presence of these tangles of adult worms and the intense chronic inflammatory reaction the immune system mounts against them over years.
This chronic inflammation physically scars and blocks the delicate lymphatic ducts. When the vessels are blocked, the lymph fluid has nowhere to drain. It relentlessly pools in the tissues of the legs, the arms, or the genitalia. This constant fluid buildup causes profound, painful swelling known as lymphedema.
Over years and decades of repeated infections, the overlying skin becomes chronically infected, vastly thickened, hardened, and horribly distorted. This end-stage progression is clinically known as elephantiasis, a morbid, grotesque enlargement of the limbs that is profoundly disabling. The textbooks features a stark, deeply upsetting clinical photo, figure 4620, showing a woman with massive, debilitating swelling of her leg, illustrating the severe physical and social toll of this disease.
Another devastating filarial nematode is Onchocerca volvulus, the causative agent of a disease known as river blindness. The vector for this worm is the black fly, a small, aggressive biting fly that specifically breeds in the highly oxygenated, fast-flowing rivers of sub-Saharan Africa and parts of Latin America.
When the black fly bites, the larvae enter the skin and develop into adult worms. The adults encapsulate themselves in dense, fibrous subcutaneous nodules under the skin, often over bony prominences. The adults themselves are relatively benign in these nodules. The devastating pathology is caused entirely by their offspring.
The adult females continuously release millions of microscopic, highly active larvae called microfilariae. Unlike other filarial worms whose larvae circulate in the blood, the microfilariae of Onchocerca migrate relentlessly through the human dermis, the deep layers of the skin.
Their constant movement and the intense immune response triggered when they die causes a horrific, unrelenting dermatitis. The itching is so severe it can drive patients to distraction or suicide. The chronic scratching and inflammation permanently damage the skin's pigment, leading to a mottled appearance clinically called leopard skin.
But the true tragedy gives the disease its name. These millions of microfilariae don't just migrate through the skin of the torso, they migrate upward, traversing the tissues of the face, and they actively invade the structures of the eye.
When they enter the cornea, the anterior chamber or the retina, and eventually die there, the immune system mounts a massive, scarring inflammatory response right across the delicate optical tissues. Over years of chronic exposure, this leads to severe corneal opacity, sclerosing keratitis, secondary glaucoma, and irreversible damage to the optic nerve. The text notes the staggering toll. Over 6 million people suffer from the severe dermatitis, and an estimated 270,000 people are completely permanently blind because of this microscopic worm's migration.
Staying with tissue nematodes, we must discuss Dracunculus medanensis, commonly known as the guinea worm. This is a parasite with a life cycle and a treatment method that sounds like something straight out of ancient mythology.
The transmission does not involve a biting insect. Humans ingest the larva by drinking stagnant water from ponds or stepwells that is contaminated with microscopic, infected copepods, which are tiny freshwater fleas. Inside the human stomach, the acid dissolves the copepod, releasing the larvae. They penetrate the stomach wall, migrate into the deep connective tissues of the abdomen, and mate.
The males die, but the fertilized female begins a long, slow migration. She grows enormously, reaching up to a full meter in length, over 3 feet long, weaving through the deep subcutaneous tissues. After about a year, when she's fully gravid and packed with millions of larvae, she migrates down to the lower extremities, usually the feet or the ankles.
To release her offspring, she must breach the skin. She secretes a highly irritating toxin that causes a burning, agonizing, incredibly painful blister to form on the surface of the skin. The intense burning sensation drives the infected person to seek relief by doing the most natural thing possible. They plunge their burning foot into cool water, like a local pond or river.
And that cooling sensation is the exact environmental trigger the worm is waiting for. As soon as the blister hits the water, it ruptures. The head of the female worm protrudes and she violently contracts, spewing a milky cloud containing hundreds of thousands of motile larvae straight into the water source. The larvae are eaten by new copepods, and the cycle continues.
What is truly fascinating, and somewhat horrifying, is the traditional ancient method of removing this worm. Because administering drugs that kill the worm while it is deep inside the tissue would trigger a massive, potentially fatal immune reaction to the 3-foot-long decaying corpse, the treatment is entirely mechanical.
You literally have to wait for the blister to form and rupture. Once the head of the worm emerges, a healer or medical worker carefully ties the tiny head to a small stick or a piece of gauze. Then they begin to slowly, agonizingly wind the live worm around the stick, pulling it out of the leg an inch or two a day. It can take weeks to extract a single worm. If you pull too quickly and snap the worm, the remaining portion retracts deep into the tissue, dies, and causes massive secondary bacterial infections, severe abscesses, and frequently gangrene. It requires immense patience and skill. Medical historians strongly believe that the universal symbol of medicine, the rod of Asclepius, which features a snake wound around a staff, actually represents the ancient, delicate extraction of the guinea worm.
It is a powerful illustration of how deeply intertwined human history and medical practice are with these parasites. Before we move to the flukes, let's briefly discuss a biological concept called larva migrans. We can think of these as the lost worms.
The text explains that the environment is full of parasites adapted to specific animals, like dogs, cats, or raccoons. When these animal parasites accidentally infect a human, our biology is close enough to theirs that the larvae can hatch and survive, but it is different enough that they cannot figure out the proper chemical signals to complete their life cycle. We are the wrong host.
Because they lack the biological roadmap, they get completely lost inside the human body and just wander aimlessly. If a human, say, walks barefoot on a beach contaminated with dog hookworm larvae and Schistosoma caninum, the larvae penetrate the skin. But they can't find the blood vessels to travel to the lungs, so they just wander randomly through the epidermis, burrowing horizontally. This creates raised, incredibly itchy, twisting red tracts visible right across the skin, a condition called creeping eruption or cutaneous larva migrans.
If the exposure involves ingesting the eggs of the dog roundworm, Toxocara canis, perhaps a young child playing in a public sandbox where a dog has defecated, the situation is much more severe. The eggs hatch, and the larvae migrate deep into the human internal organs. This is visceral larva migrans.
The larva wanders through the liver, the lungs, and the brain, causing persistent hypereosinophilia, liver enlargement, and fever. Most devastatingly, they can enter the globe of the eye, causing ocular larva migrans. The immune system forms a large, damaging granuloma around the trapped worm on the retina. It severely damages vision and, tragically, presents as a solid mass that is frequently misdiagnosed as retinoblastoma, a type of eye cancer, sometimes leading to the unnecessary surgical removal of the child's eye. It underscores the vital public health importance of rigorously deworming our domestic pets.
Let's look at the final major group of helminths, the trematodes or flukes.
Flukes have a very distinct morphology. Unlike the cylindrical roundworms or the ribbon-like tapeworms, flukes are generally flat, fleshy, and leaf-shaped. And almost universally, their life cycles are incredibly complex and absolutely require a very specific intermediate host to undergo necessary developmental stages. A freshwater snail.
For most flukes, like the Chinese liver fluke, Chlorocos sinensis, or the sheep liver fluke, Fasciola hepatica, the larvae develop in the snail, swim out, and insist themselves on aquatic plants or in the flesh of freshwater fish. Humans become infected by eating these raw plants like watercress or raw fish containing the encysted larvae.
But the textbook highlights a massive, highly dangerous exception to this rule of ingestion, the Schistosoma species, collectively known as the blood flukes. They completely bypass the digestive tract. They do not wait to be eaten.
Schistosomes are unique trematodes. When their highly motile, fork-tailed infective larvae called cercariae are released by the millions from the snail into fresh water, they actively hunt for a mammalian host. If you are wading, swimming, farming, or washing clothes in infested water, the cercariae detect the lipids on your skin. They attach, drop their tiny swimming tails, and secrete enzymes to directly penetrate your intact skin within minutes.
Once inside, they enter the venous circulation. They migrate through the lungs to the liver, where they mature. And here's another unique feature. Unlike all other flukes, which are hermaphrodites, Schistosomes have highly distinct separate male and female sexes, and their physical pairing is fascinating.
The textbook has a remarkable microscopic picture, figure 4623, showing an adult male and female worm in copula. The male worm is larger, thicker, and has a deep groove running longitudinally down the entire length of his body, called the gynecophoral canal. The female worm is much longer, thinner, and darker. She rests entirely inside this groove, completely embraced by the male.
Once they find each other, they mate for life. The male physically carries the female against the blood flow, migrating out of the liver to their final destination, the complex venous networks lining the human organs. Schistosoma mansoni and Schistosoma japonicum specifically migrate to the mesenteric veins surrounding the large and small intestines. Schistosoma hematobium migrates to the dense venous plexus surrounding the urinary bladder. They can live locked in this embrace inside your blood vessels for up to 20 or 30 years.
Surprisingly, the adult worms themselves cause very little immune response or direct damage. They actively disguise themselves by absorbing human blood proteins onto their surface, essentially becoming invisible to the immune system. The catastrophic pathology of schistosomiasis is caused entirely by their eggs.
The female lays hundreds to thousands of eggs every single day. The worms are living in the veins outside the organs, but the eggs need to get inside the gut lumen or the bladder to be passed out in feces or urine. The eggs are equipped with sharp terminal or lateral spines. They secrete olytic enzymes to physically dissolve the blood vessel walls and push their way through the dense tissue of the intestines or the bladder.
It is an incredibly inefficient process. At least half of the eggs never make it out. They are swept backward by the venous blood flow and become permanently trapped deep in the tissues, lodged in the walls of the intestines, trapped in the bladder wall, or swept deep into the liver.
The human immune system easily recognizes these trapped spined eggs as highly foreign objects and mounts a massive, unrelenting inflammatory response. White blood cells swarm the eggs, forming huge, dense, fibrous structures called granulomas around every single trapped egg.
Over years and decades of thousands of eggs being laid daily, this massive inflammatory response fundamentally destroys the architecture of the organs. In the liver, the millions of granulomas cause profound fibrosis, massive scarring. This scarring chokes off the blood flow through the liver, causing severe portal hypertension. The pressure in the veins backing up from the liver becomes so immense that the veins in the esophagus can balloon and rupture, leading to fatal internal bleeding. The pressure forces fluid out of the vessels, completely filling the abdominal cavity in a condition called ascites, giving patients a severely distended belly.
The pathology for schistosoma hematobium in the bladder is equally grim. The chronic, decades-long inflammation, the constant mechanical irritation from the spined eggs, and the severe tissue damage cause profound hematuria of blood in the urine. More terrifyingly, this specific chronic irritation is directly causally linked to cellular mutations. Schistosoma hematobium is one of the few biological organisms definitively proven to cause cancer, specifically squamous cell carcinoma of the bladder.
It is a monumental global health burden, affecting over 200 million people worldwide, intimately tied to agricultural practices and water use. Which leads us to our final section of the dive. We are looking at the tissue cystodes. I promised you an aha moment at the beginning of this episode, a massive conceptual shift that redefines how we look at an organism we already discussed. And the textbook delivers this perfectly regarding one specific parasite, the pork tapeworm, Taenia solium.
This is perhaps the most vital distinction to understand in the entire field of medical parasitology. We discussed tapeworms earlier, like the fish tapeworm, living comfortably in the human gut. The standard life cycle of Taenia solium works the same way. The pig is the intermediate host. The pig eats eggs from contaminated soil. The larvae hatch, migrate into the pig's muscle, and form a dormant cyst called a cysticercus.
If a human eats that undercooked pork containing the cysticercus, the cyst survives the stomach acid. The tiny tapeworm head pops out, attaches to the human intestinal wall, and begins to grow into a massive adult Taenia solium tapeworm.
And as the text notes, harboring an adult pork tapeworm in your gut is usually a relatively mild asymptomatic condition. The adult worm doesn't invade the tissues. It simply absorbs nutrients and sheds its egg-filled segments, the proclodids, out into the human's feces to continue the cycle. The human is the definitive host. The worm is where it belongs.
But here's the critical pivot, the scenario that changes everything. What happens if a human doesn't eat the undercooked pork? What happens if instead a human accidentally ingests the microscopic eggs directly from human feces? Imagine a scenario where a food handler harbors an adult tapeworm, fails to wash their hands properly after using the restroom, and prepares a salad.
In that scenario, the entire biological dynamic shifts. You bypass the adult worm phase completely. When you swallow the egg, you are acting as the intermediate host. You are acting like the pig.
The eggs hit your stomach and the acid dissolves the shell. They release tiny, highly invasive larvae called oncospheres. These larvae do not stay in the gut. They treat your body exactly like they would treat the pig's body. They use sharp hooks and enzymes to physically bore through your intestinal wall, enter your bloodstream, and ride the circulation into your deep tissues to encyst.
They seek out highly oxygenated areas. They form expanding, fluid-filled cysts inside your skeletal muscles, inside the tissues of your eyes, and most dangerously, deep within the tissue of your brain. This systemic infection is called cysticercosis, and when it involves the brain, it is called neurocysticercosis.
The textbook includes an MRI scan, figure 4618, that is incredibly sobering. It shows a cross-section of a human brain absolutely riddled with dozens of these dark, fluid-filled cysts. Inside every single one of those cysts is a tiny, inverted tapeworm head just sitting dormant in the brain tissue, waiting to be eaten by a predator that will never come.
As long as the cysts are alive, they actively suppress the local immune response. But eventually, the cysts begin to die and leak fluid. The brain's immune system detects this highly foreign protein and launches a massive inflammatory attack. The swelling and pressure inside the closed skull cause severe intractable seizures, focal neurologic deficits, hydrocephalus, and frequently death. Neurocysticercosis is an incredibly major leading cause of adult-onset epilepsy worldwide, particularly in regions where pork is consumed and sanitation infrastructure is poor.
It is a profound realization. It is a completely different, infinitely more severe, life-threatening disease caused by the exact same parasite, entirely depending on one single variable, whether you ingested the dormant cyst embedded in meat or the microscopic egg shed in feces.
We see a very similar, highly dangerous pattern involving humans acting as accidental intermediate hosts with our final parasite, Echinococcus granulosus, the causative agent of hydatid cyst disease.
The natural life cycle of this tapeworm does not involve humans at all. It cycles strictly between canines, typically dogs or wolves, and herbivores, typically sheep. The adult tapeworm is tiny, only consisting of 3 segments, and it lives safely and harmlessly attached to the intestinal wall of the dog. The dog passes the eggs in its feces onto the pasture. The sheep inadvertently grazes on the contaminated grass and eats the eggs. The eggs hatch, the larvae bore into the sheep's liver or lungs and form cysts. The cycle completes when a farm dog or a wolf kills the sheep or is fed the infected offal, eating the cysts.
Humans enter this cycle strictly by accident. If a human ingests the microscopic eggs from dog feces, perhaps from petting a working farm dog that has rolled in contaminated dirt and licked its fur, and then the person eats without washing their hands, the eggs hatch in the human stomach. The larvae penetrate the human gut, enter the portal vein, and are typically trapped by the first major organ they hit, the liver. If they pass the liver, they lodge in the lungs.
Because the human immune system cannot kill them, and because they are not in the correct host, they do not form tiny, dormant cysts like they do in the sheep. They grow uncontrollably. They form massive, expanding, fluid-filled structures called hydatid cysts. The text notes these cysts can grow up to 7 centimeters or more in diameter every single year. They can easily reach the size of a grapefruit, a melon, or even a football, slowly, relentlessly compressing, starving, and destroying the healthy liver or lung tissue around them.
The visual evidence in the book is astounding. Figure 4624 shows a massive 14-centimeter cyst surgically removed from a patient's spleen. It looks completely alien, a massive taut balloon of tissue, and the interior of that cyst is terrifying. It is filled with toxic fluid, and budding off the inner wall are thousands upon thousands of protoscoluses, tiny, infectious, fully formed tapeworm heads, essentially acting as seeds, just floating in the hydatid fluid.
The presence of that fluid brings up a terrifying, highly specific surgical complication. If a patient requires surgery to remove one of these massive cysts from their liver, the surgeon must be incredibly careful. If the scalpel slips, or if the pressure causes the cyst wall to accidentally rupture during extraction, that highly immunogenic, toxic fluid spills directly into the patient's open abdominal cavity.
The host's immune system, which has never been exposed to these specific internal cyst proteins before, reacts so violently and instantaneously to that massive antigen spill that it triggers catastrophic systemic anaphylactic shock. The patient's blood pressure drops to 0, their airway closes, and it can be immediately fatal right there on the operating table.
Furthermore, if the patient survives the anaphylaxis, the nightmare isn't over. Any of those thousands of surviving microscopic tapeworm heads that spilled out into the abdomen can individually seed themselves into the surrounding tissue. They will attach to the peritoneum, the bowels, the remaining liver, and each individual head will begin to grow into a brand new, massive, hydatid cyst. A single surgical error can metastasize the parasite throughout the entire body. The surgical protocols require meticulously isolating the cyst and often carefully injecting strong parasiticidal chemicals directly into the cyst to kill everything inside before even attempting to remove it.
The level of caution required highlights just how incredibly potent these organisms are, even when they are trapped in the wrong host.
We have covered an unbelievable, monumental amount of ground today. From the single-celled hitchhikers causing explosive outbreaks at luxury ski resorts because of a failing water filter, to the blood flagellates playing a genetic game of chess with our immune system, to giant foot-long worms taking a mandatory, destructive detour through human lungs just to grow up. The scope of biological engineering here is vast.
The overarching question for anyone studying this material or listening to this is, why does understanding this deep biology matter? It matters because understanding these complex, sometimes convoluted life cycles, understanding their mechanical vulnerabilities, is the absolute key to breaking them. We saw that principle repeated time and time again in the textbook.
You disrupt the cycle at any point, and you defeat the parasite. That means investing heavily in modern sanitation infrastructure to completely sever the fecal-oral transmission routes. It means establishing rigorous food safety protocols, teaching people to thoroughly cook their meat to exact temperatures to destroy those hidden tissue cysts. It means utilizing vector control, distributing simple insecticide-treated bed nets to physically separate humans from the Anopheles mosquito, or clearing dense brush near rivers to eliminate the tsetse fly habitats. And it means constantly testing, rigorously treating, and multi-filtering our public water supplies to catch those incredibly resilient cysts. Clean water, safe food, and basic hygiene are the greatest, most effective shields humanity has ever built.
The material we discussed today isn't just academic. While the life cycles might sound like pure science fiction or body horror, it is very real functional biology that impacts the daily lives, the economies, and the futures of billions of human beings across the globe. Whether you are traveling internationally on business, whether you are adopting a new puppy that hasn't been dewormed, whether you are going backpacking in the local mountains, or even just buying fresh imported raspberries at the grocery store, this knowledge is intensely practical. It directly informs how we interact safely with the natural world.
I want to leave you with a final lingering thought to chew on as we wrap up. Something the text constantly points toward but leaves for us to deduce. If these parasites have evolved such insanely complex life cycles, if they can dynamically change their genetic codes on command to outsmart our highly evolved antibodies, if they can secrete precise chemicals to literally reprogram human muscle DNA to build them a house, if they know exactly how to surf our bloodstream to migrate through multiple organ systems just to end up in the exact right place, what does that say about the evolutionary arms race happening right inside us? We often think of humans as the absolute pinnacle of biology, mastering the world around us, but looking at the sheer sophisticated ancient engineering of these parasites, a question arises. Are we shaping the parasites through our medicine, or are these parasites fundamentally permanently shaping human biology and evolution? Who is really driving the evolutionary car?
That is a profound question. When you look at the genetic adaptations humans have developed just to survive malaria, you realize how much they have steered our development.
It changes your perspective entirely. Thank you all for joining us on this incredibly intense journey through Chapter 46. Stay curious, stay informed, and honestly, please double check that you cooked your pork all the way through tonight. We'll see you on the next deep dive.